This figure is showing what the previous figure referred to as the mitotic phase, which is mitosis followed by cytokinesis. There's a tremendous amount of detail here that we're not going to be talking about. So what I'd like to point out to you are the phases that I would like for you to know. So we're going to eliminate the pro-metaphase stage because that's really not relevant. So we're going to say prophase, metaphase, anaphase, telophase. So those are going to be the major stages of mitosis. And what I want you to realize is what's going on, or recognize is what's going on with the chromosomes during that particular phase. So prophase, the cell's DNA has replicated during S phase of interphase, and what now begins to happen is the chromosomes begin to condense, and they're wrapping around proteins, and we talked about this earlier, histone proteins they wrap around, and you're taking, in a human cell, about 2 meters of DNA, and it's being condensed down to an incredibly small size. Or look at the bottom scale, which is showing a fluorescent image of the process of mitosis. This line is representing a scale of 5 micrometers. So the whole diameter of the cell is about 10 micrometers. Remember, micrometer is a millionth of a meter. So we're talking about incredibly small sizes here. So the nuclear membrane starts to break down, the chromosomes are condensing. We're going to shift over to metaphase. When you see meta, think middle. So what happens is the duplicated chromosomes align along the equator or the center of the cell. Now you can predict what's going to happen because remember, each daughter cell needs to get one of the sister chromatids from each duplicated chromosome. So when they line up along the equator, one chromatid from each chromosome is going to go one direction, the other chromatid would go the other direction. So again, they're lined up and fibers are attached to the centromeres to help pull those replicated portions of the chromosomes called the sister chromatids apart from each other. Anaphase, the sister chromatids have separated from each other and they're beginning to be pulled by these fibers to the opposite poles of the cell. In telophase or telophase, the chromosomes have reached the opposite pole, the nuclear membrane begins to reform, and the chromosomes begin to decondense. Cytokinesis, as we mentioned earlier, is the actual separation of the cytoplasm. We're focusing only on animal cells. So what happens is a cleavage furrow forms. In other words, think about it this way. If you take a balloon, one of the kind of rectangular shaped balloons, and you don't fill it up completely with air to where it's really, really tight, you can take it and you can twist it in the middle, and you can twist it so that in essence pinches it into 2 parts. That's what you can think of cytokinesis as happening. The membrane is actually cleaved and 2 separate cells form. So again, I'm asking you to know prophase, metaphase, anaphase, telophase, and where you would see the chromosomes in each of those positions. And then obviously cytokinesis, when the membrane itself is actually being pinched into 2 parts to create the 2 daughter cells.